what I'm going to try to do today is to talk to you about the path that I took to get to the Nobel Prize. And I'm going to talk about a lot of things that were very influential in my life and that I learned lessons that I think are very valuable for many students. So I will talk about those as we go along. And at the end, I will summarize some of those important things, which I think are really valuable lessons for all students. So I was born in the town of Derby, England, in the middle, it's right in the middle of the country on the left-hand panel of this slide. And I was born during the Second World War, 1943. And at that time, there was a Rolls-Royce factory that was making aircraft engines very near to Derby. And the Germans were constantly trying to bomb that factory. And they flew right over Derby and often dropped bombs not in the correct place. And the street on which I lived, one side of it had some quite severe bomb damage, but not the side I lived on. And so this was the first real demonstration of my luck. And I think you will see during the course of this talk that I am one of the luckiest people you know. So here I am, living in Derby in a war zone, but my street didn't get, or the side of the street that I lived on didn't get hit. When I was 4 years old, I moved to Bath. My parents moved there. My father was a motor mechanic and got a slightly better job working for the council in Bath. That's shown on the right-hand panel. And it's about 100 miles due east of London. It's a very beautiful town. And I think if any of you ever get the chance to go to England, apart from visiting London, you should really seriously consider visiting Bath. Now, when I was young, I went to a small school close to where I lived, and then to what they call a junior school in England from age 7 to 11. It was a very small school. It's called St. Stephen's School. It's shown here. And I learned a number of things during my time at that school. But one of the most interesting was that I fell in love with mathematics. And that happened because at the school, when I used to leave in the evening after school was over, the headmaster would often give me little puzzles to do, little mathematical puzzles. And I used to take them home, and I discovered that I could usually manage to do them. And they got progressively harder. And it wasn't until I was probably about to leave the school that I realized I was the only kid that he was giving these puzzles to. And I think he must have seen something in me that I certainly was not aware of at the time. But nevertheless, it really got me to love mathematics.
Dear Professor, I'm sorry, but we still see the first slide.
Oh,
probably
really?
You are changing the slides. But...
Okay, so let me go back.
Maybe it's better to stop sharing and start once again.
Okay.
And share the full screen mode, or share the entire screen.
Oh, this is very annoying.
Well, I can stop it for you, and you will
I'll stop it and start again. Would it be better if I sent you the slides and you could then use them?
Yes, we can, but you can try now to share again.
Okay.
And just leave the entire screen when you share.
Dear Professor, you can send the slides and at the same time try to share from the very beginning.
Okay, let me do that.
I think if you go to presentation mode, you might well be able to get it on.
Okay, so I've sent the slides to you. Now, what can you,
Okay.
So-
I will check.
is there something I can do here?
Just go down to the very bottom and
Yep.
press presentation mode one, which is notes, and then there's a couple of other. That's it. Just try that. See if that works. Now the next one on that one. Just try that again. There you go.
Yes, now it's in the full screen mode, and probably if you change the
That's perfect.
slides, we'll see it.
You click
Okay,
on.
let's see.
Yep, fine.
Yes, it's
Okay. Mhm.
Okay, fine. So rather than go through the lengthy repeat, I'll try and do it very briefly. So I was born in Derby, shown on the middle of the left-hand panel. It's in the middle of England. And where I grew up was on the flight path for the German bombers trying to destroy the factory where Rolls-Royce were making fighter engines. When I was 4 years old, moved to Bath. My father got a better job. And I mainly grew up in Bath. Bath was really where I consider that I grew up. This was the junior school that I went to, a very small one. It was 4 classes for 4 different grades, and there were about 30 students or so in each class. And that was where the headmaster used to give me puzzles to do at night, really little mathematical puzzles, and I fell in love with mathematics. But I also fell in love with a lot of other things while I was there. By age 11, I really wanted to become a detective. I read a book entitled How I Became a Detective, a Traditional Detective in the Police Department. And that sounded to me as though it would be a lot of fun. And I think now that I do research, I realize that wanting to become a detective and traditional police work is really all about solving problems. And research is the same. So I always put that down to the fact that it led me eventually to research. The pictures are pictures of the city of Bath. On the bottom left are the Roman baths. Bath is a very old city. It was developed originally by some locals who lived in England who noticed there were hot springs nearby and built their settlement near it. And then the Romans came and made a professional bath out of it. And so these Roman baths are really very nice, and they're right in the center of the city. The picture on the right is one of the crescents that was built. Actually, it's quite close to where I used to live, although I just lived in a council flat a little way away. But again, it's beautiful Georgian architecture that really makes Bath a very special place to go and visit. Now, when I was in Bath, I really had very good education. I got into one of the classes where they really made all the students, starting at about age 6 or 7, really talk to the rest of the class about anything that they had been told to read the night before. And so we really learned quite a lot about how to make presentations, but to do it by avoiding a lot of technical language that might confuse people who were not in your field. When I was 11, my father bought me a chemistry set, one of these really little primitive chemistry sets that they give to children. And I soon decided that it was possible not just to do all of the rather simple and I might say trivial experiments that came with the chemistry set, but it was also possible to make fireworks and explosives. And so this is where I really came to love chemistry. I was absolutely overwhelmed by it. My father had a friend who worked in a pharmacy nearby, and he used to get any chemicals that I wanted. And my father also built a tiny chemistry lab, just a cabinet with a formica top in the kitchen where we lived. And I used to do all sorts of interesting chemical experiments. Now, as I got older, I went to this school, the City of Bath Boys School, a so-called grammar school. It was the high school in Bath and the best that there was there. I had a very good time. I just absolutely continued to love mathematics. I learned to play the violin and joined the school orchestra, and I got a lot of fun from that. I was also rather close to a limestone escarpment called the Mendips. You probably have heard of cheddar cheese, and it came from a town, Cheddar, in the Mendips. And because it was limestone, there were caves everywhere. And I used to enjoy caving. I went down into the caves and my friend and I would collect bugs that lived in the caves and send them off to a collector who would identify them for us. I also discovered jazz, for which Bath has now become quite famous. They have a jazz festival every year. Now, I did chemistry when I was at school, but in fact, it turned out that the chemistry teacher and most of what he was teaching was stuff that I already knew, because as a result of having this chemistry set at home, I'd gone down to the local library and used to take out books on chemistry and was really way ahead of anything that I was being taught in school. So that was not such a good experience. A lot of the chemistry I find kind of boring. The other thing we had to do at school was to take physics, which you needed in order to get into university if you wanted to do science when you were there. And we had a terrible physics teacher who I didn't like very much, and he taught physics in a way that I didn't appreciate. And eventually, at age 18, when we had to take the advanced level exams to see whether we could get into a good university, I actually failed physics. And that meant that I couldn't go to university when I was 18. I had to wait a year until I was 19. And then I went to Sheffield University. I did pass physics, but it was one of these things that it was the way that the subject was taught that was bad, not the subject itself, because as soon as I got to university, I discovered that physics was really all mathematics. And of course, since I love math, then that was all very nice. So that worked quite well. I went to Sheffield University because since I failed physics the first time around, I couldn't get into Oxford or Cambridge, which was the sort of natural university to try to go to. And I also wanted to do chemistry, and the teaching of chemistry in both Oxford and Cambridge was not as good as it was in two of the other universities in England. One of those was Southampton in the southern part of the country, and the other was Sheffield, just north of Derby. I applied to both. I got into Sheffield. I was not accepted into Southampton, but Sheffield turned out to be probably the best place I could have gone to. Looking back, I had such a good education there. I had a really good time. I very much enjoyed being there. I had very good chemistry taught while I was there. And I got a bachelor's degree in chemistry, not a first-class degree, a second-class degree. But nevertheless, it was good enough to allow me to stay on and become a graduate student and do graduate work. And from there, I moved from doing general chemistry into organic chemistry. I was given a piece of wood from a Brazilian tree and told to find out what was in it. And that was kind of fun. And it turned out that during that time, I really found that the most important thing was to have a good mentor, someone who would really take an interest in you, help you, and make sure that you did things properly. And I was fortunate in Sheffield that this gentleman from Japan, Kazu Kurosawa, worked in the lab that I had been assigned to. And he was an extraordinary person. He was a postdoc at the time, and he really took me under his wing. He showed me how to do experiments, what experiments to do in order to really make progress and to get all of the results I needed for a PhD. And he did so not just by telling me what to do, but explaining to me why I was doing it, and then to help me to understand the results that came. And this was, I say he played an incredibly important part in my life. I didn't appreciate it as much at the time as I did afterwards, but thanks to him, not only did he teach me chemistry, he also taught me how to play Go, this game on the bottom left. And actually, after he left to go back to Japan, we played Go for quite a long period of time. He also taught me how to play shogi, which is the Japanese version of chess. And he was just generally always there for me and always helpful. Now, I got all of the results that I needed, thanks to Kazu, within the first year of my PhD. And I could have written my thesis right then. But in England, you had to spend 3 years doing a PhD. And Kazu had gone at that time. At the end of my first year, he went back to Japan. And so I had to find other things to do. And so I started to do some experiments that looked as though they might be biochemistry, trying to find out about an interesting compound that I discovered called a neoflavonoid. And it was really something that I think a good biochemist could probably have helped me to do, but I tried to do it on my own and failed miserably. By the end of the second year, I'd started to look around and think what I was going to do after my PhD. And I spent a lot of time in the library at Sheffield. And Sheffield had a very, very good library with all sorts of good science books. And I noticed that there were something called books about molecular biology. There was a Journal of Molecular Biology. And then I came across this book called The Thread of Life by John Kendrew. John Kendrew was a molecular biologist and an X-ray crystallographer. He was one of the first people to get a structure for a protein. And what he described in this book was really the origins of molecular biology. He talked about the first individuals who got it going, talked about the molecular research lab that took place in Cambridge, the MRC lab in Cambridge. And by the end, when I finished that book, I really felt that molecular biology was what I wanted to do. And it became an obsession in my mind. I didn't want really just to do chemistry anymore. I wanted to do molecular biology. But it was at a time in 1965, 64 time, when molecular biology wasn't really recognized as a subject by a lot of people, including everybody in the chemistry department at Sheffield, except for one man, Mike Blackburn, who was a young assistant professor. And he was the only one who really encouraged me to pursue a career in molecular biology. So anyway, thanks to prompting from him, I then went on and applied to 6 labs that actually were all chemists on the brink of molecular biology. So they were doing biological chemistry, I would say, rather than perhaps molecular biology. I applied to all 6, and only one accepted me. And that one was Professor Jack Strominger. And when I applied to him, he was a professor in Wisconsin, right in Madison, Wisconsin, right in the center of the USA. And so I thought I would be going there for a postdoc. But just before I was scheduled to leave, he wrote and said, could I delay for a couple of months, because he'd just been promoted to be a full professor at Harvard, which turned out to be his lifelong ambition. And so I waited for a couple of months, and then with my family, we moved to the US, to Cambridge, just north of Boston, right next to Boston, and I became a postdoc at Harvard. And while I was there, the project I was given was to finish up a project that another postdoc had started who'd worked with Jack Strominger in Wisconsin. His name was Tom Stewart. And it was to find the sequence of a transfer RNA that was involved in bacterial cell wall biosynthesis. Jack had made a career of understanding how bacteria laid down their cell walls. He'd been incredibly successful at this, but during the course of this, he discovered that there were cross-links in the peptidoglycan that made up the bacterial cell wall. And those cross-links often had amino acids in them. And in particular, in this organism, Staphylococcus epidermidis, there was a glycine, a string of 5 glycines were in the cell wall, and those glycines were donated by this tRNA. And the tRNA was interesting because it turned out it only worked in bacterial cell wall biosynthesis. It didn't work in protein synthesis. And the question was why? What was different about this tRNA that made it work in cell wall biosynthesis? So Tom Stewart had begun by trying to sequence, well, first of all, to isolate and then sequence the tRNA using what we would now think of as very traditional methods, which were methods that had been pioneered by Bob Holly, who had won the Nobel Prize for it. However, that method was very slow, it was quite tedious, and the more I read, the more I realized that there were better methods being developed. And one of those was being developed by a man called Fred Sanger, who was at the MRC lab in Cambridge, and who had previously won the Nobel Prize for sequencing proteins. And he had turned his attention to how to sequence RNA. And so he was using P32 labeled RNA rather than the unlabeled tRNA that Holly had been using. So I wrote to Fred Sanger and asked if I could become a go and spend some time in his lab and learn how to do it. And he wrote back and said, yes, please come to the lab. And so I went and spent 6 weeks in Fred Sanger's lab. He's shown underneath the red arrow here. And he, in fact, went on to get a second Nobel Prize later in 1980 for DNA sequencing, which he shared with Wally Gilbert. Now, when I first arrived at the MRC lab in Cambridge and went to see Fred, I'd gone into the lab on a Sunday because one of his technicians had told me how to go in and get set up. And I go into the lab and there's this little man there who is washing the dishes. And I thought it was the janitor. And so I introduced myself and I said, I'm Rich Roberts. I'm here to work with Fred Sanger. And he looked at me and said, oh, I'm Fred Sanger. Here was a Nobel Prize winner washing dishes on a Sunday. And it was absolutely typical of Fred. He is probably the most modest scientist I've ever come across. Really nice guy. But very humble, very modest, never trying to tell you how good he is. And for me, he's really become something of a role model in science. He, for me, typifies what scientists should be, just doing good work, but being modest about it and not spending all their time telling you how smart they are. So anyway, I spent 6 weeks there, went back to Harvard, and at that point, Jack built a small lab that would emulate the sequencing facility that they had in Fred Sanger's lab so that we could do, and I in particular could do exactly the same thing I'd been doing there. And by chance, it turned out that during that period of time, I was the first person in the Boston area to be doing RNA sequencing using Fred Sanger's methods. And so that turned out to be really very good because a lot of people came to me to learn how to do it, even though I'd only just learned how to do it myself. And so I met a lot of people, including Wally Gilbert, the person on the left here, who at that time was busy sequencing RNA and working on the lac repressor. There were many others who came too, and I made a lot of friends in that period who turned out to prove extremely useful and very helpful. Now, when I finished my postdoctoral period in 1972, I had to look around for a job, and I really wanted to come back to England. The only reason I'd gone to abroad in the first place was because if you wanted to get a job in England, an academic job, you really had to go abroad for a postdoc and then come back. I applied for a job in Edinburgh, which I had been told I was perfect for, but I never heard back from them. And in the meantime, I got 2 offers, one from Jim Watson of double helix fame, and another one from the biolabs, George Wald in the biolabs at Harvard. And they both offered me a job there. And I had a wife and 2 children, and I needed a job. I needed some income. And so one day, Mark Potashny of Lambda repressor fame came to me after a seminar and said, oh, Jim Watson has a position for you that would be perfect at Cold Spring Harbor, and he's going to come and talk to you about it. Well, after a couple of weeks, I never heard anything, and Mark came through and he said, well, Jim Watson doesn't actually know who you are, so perhaps you could go and introduce yourself. So I did. I went and introduced myself, and basically, almost as soon as I got in the office, Jim offered me a job, and then didn't really conduct what I would consider a proper interview. But as I was leaving his office, he said, oh, you probably should come down to Cold Spring Harbor and visit, but the job is yours. So anyway, I did. I went down to Cold Spring Harbor, I visited, and at that point, I realized that this was a very nice opportunity for me. And the job that he had in mind was to sequence the DNA of the SV40 virus. This was one of the first tumor viruses that had been found. Sounded okay, because the way you sequence DNA those days was to copy it into RNA and then sequence the RNA, and I knew how to do that thanks to my visit to Fred Sanger's lab. Anyway, so I went down, and as soon as I got there, I discovered there were two other groups in the world also already sequencing SV40. And it seemed to me silly to be doing the same thing, and certainly silly to do it by the same method, that was to copy it into RNA and then just sequence the RNA. But fortunately, for me, I had attended a seminar when I was at Harvard before I left by this man, Dan Nathans, the top left figure, in which he described the discovery of restriction enzymes and what one could do with restriction enzymes. Actually, just one restriction enzyme, endonuclease R, which I had heard about. And that had been discovered by Hamilton Smith, the person in the bottom left-hand corner, and the three of them, with Werner Aber, shared the Nobel Prize in 1978 for this discovery of restriction enzymes. And it occurred to me when I got to Cold Spring Harbor that there were several other restriction enzymes that had been discovered. And I thought, well, maybe there's more. Maybe there are some, and maybe we could use these in order to develop sequencing methods for DNA. Because the reason that RNA sequencing methods came along first was because RNA, there are many small molecules, tRNA, 5S RNA, and so on, lots of small molecules to practice on. And if you want to develop a technology to do something, you need something small to practice on. There were no small DNA molecules. And so it occurred to me that I could use these restriction enzymes to make small DNA molecules, and perhaps then to develop novel methods for sequencing DNA. I have to tell you that Jim Watson didn't like this at all. He said, you know, I hired you to sequence SV40, get down there and do it the way these other groups are doing it. And so we got off on the wrong foot. And actually, I was 20 years at Cold Spring Harbor. And for most of the time, I ended up fighting with Jim Watson because he had very clear ideas of what he thought I should be doing. But I really don't like people telling me what to do. I like to do the things that I've thought of. So anyway, to cut a long story short, I started looking at these restriction enzymes, and everywhere we looked, we found there were more. And in the early 1970s and early 80s, 75 of the first 100 known type 2 enzymes were isolated in my lab at Cold Spring Harbor. This led to something interesting. I met Norton Zinder, who was also interested in restriction enzymes. We actually met on a train going to a meeting in Belgium. And he became a very good friend, and also someone who proved incredibly helpful in my career. He was probably Jim Watson's only really good friend. And Norton liked me. And whenever Jim Watson threatened to fire me, which he did on a number of occasions, Norton would step in, talk to Jim, and persuade him that I should be kept on. So he played another important role in my scientific life. Now, while we were isolating all of these restriction enzymes, I had a number of postdocs coming in. And the postdoc, the first project was always to look for a new restriction enzyme because we knew we were going to find them and that you would get a publication out of this. So this was sort of what I think of as a bread and butter project for a postdoc, where you knew you were going to find something, and at this time a new restriction enzyme was worth a publication. But then the postdocs always, we had to find another project for them to do. And in 1974, Richard Gelinas, who I'd met when I was at Harvard, he'd been a graduate student at Harvard in Fotis Kafados' lab, and he applied to become a postdoc, and I encouraged him to do so when I was at Harvard. But anyway, he came down, and he was interested in working on adenovirus 2 messenger RNA. And the project was a very simple one. It's outlined at the bottom. And at the time, we knew what a prokaryotic promoter looked like because, well, Wally Gilbert and Mark Potashny had already isolated them and looked at them. And the question was, do prokaryotic promoters and eukaryotic promoters look alike? Are they the same? That is it the same signal that the RNA polymerases are recognizing? And we decided we would do this in adenovirus because this was a virus that a lot of people at Cold Spring Harbor were working on. It was more complicated than SV40, and it was a linear virus. And we knew from work that had been done in Joe Sandbrook's lab, Phil Sharp among them, and we had worked out how to map adenovirus 2 DNA using the restriction enzymes. And that actually is the only publication I've ever had with Phil Sharp, the co-publication. But we were interested in the sequence of adenovirus, and particularly the sequence at the end, because it turned out that early after infection, transcription starts at the very end of the DNA. And the thought was that if we could map these very end pieces and sequence them, find out where the messenger RNA started, and we would do this by looking at the extreme 5' end of the messenger RNA. And I developed some methods for doing this that we thought were pretty good, but we didn't get the results we were expecting. What we found was that in early messenger RNA, we couldn't get enough messenger RNA to do experiments. And so we had to start working on late messenger RNA. And late in infection, almost all messenger RNA synthesis is of adenovirus messenger. And so there was loads and loads of messenger RNA. And at the time, we were looking for the 5' end of the messenger RNAs, of the late messenger RNAs. And basically, we were doing this by looking for a triphosphate oligonucleotide that we could get with ribonuclease T1. Ribonuclease T1 cuts all the RNAs after G residues. And so we thought there should be a triphosphate and then a short oligonucleotide that was the 5' end. But while we were doing this work, caps were discovered. And so now we started to look for caps on the 5' end of messenger RNAs. We developed a method, a very nice method to do this, because that actually made it easier than finding the triphosphates. And so we looked at adenovirus late messenger RNA, and we discovered that although we knew there were some 15 or 20 different messenger RNAs, they all had exactly the same cap on them. It was an 11-long nucleotide, oligonucleotide, and it didn't matter which messenger RNA you looked at, they all had the same. And the question was, how is this possible? Because late during infection, there's a major RNA is made starting at about 17% of the way along the genome. But then there were all these others, 10, 12 others at least, maybe 15 others, but they all had the same cap. Well, we spent a lot of time trying to work out what was going on, and we were able to show quite clearly using basic biochemical experiments that a messenger RNA that came from maybe 3/4 of the way along the genome had the main body of its message, encoded at that position in the genome, but that the capped oligonucleotide attached to it came from a position elsewhere in the genome. And the question was, how did these get together? What was going on? This was the paper that described the original discovery of these capped oligonucleotides, but we didn't have a good explanation. We had a few possibilities. And then eventually, after quite a long period of time, in 1977, I was sitting in the office with Richard Gelinas one day on a Saturday morning. And we usually had gotten into this habit of talking about why last week's experiments hadn't shown what we wanted, and that the current set of experiments, let's come up with some new ones and see how we can prove what is going on. Well, Richard was explaining some very complicated experiments he wanted to do, and it suddenly struck me, a thought came to me about how we might show what was going on. And this was best if you look at the top right-hand panel. The idea was that we would make an R loop between the DNA and the RNA, and we knew that it was possible to do this. And when you hybridize RNA, say messenger RNA, to double-stranded DNA, the resulting RNA-DNA hybrid forms a loop, because RNA-DNA hybrids are stronger than DNA-DNA interactions. Now, in adenovirus 2 messenger RNA, we knew at the 3' end there was a poly A, because all messenger RNAs had poly A at the end. And at the left-hand side, at the 5' end of the RNA, we knew this oligonucleotide that we'd found was there, but that it wasn't coded right next to the message, the main body of the message. So the idea was, maybe we could find a DNA that did code for it, and that we could then add this single-stranded probe DNA to the R loop, and we would get hybridization, and we would know where this was being coded. We didn't actually know where it was at the time, although I had a good guess of where it was being coded. And so we made the appropriate DNA. Now, I thought of this experiment on a Saturday morning, and Richard went on and made the reagents that we needed to do it. But unfortunately, this was an electron microscopy experiment, and neither Richard nor I knew anything about electron microscopy. But fortunately, these two individuals, Louise Chow and Tom Broker, who were very good electron microscopists, knew how to do this experiment. And in particular, Louise Chow did the experiment. She was really very, very good at this. We had made the reagents by the Tuesday, and on Tuesday morning, she did the experiment. And what she found was not what we were expecting to see in the middle, but rather she found what we saw at the bottom, that not only did the probe DNA we'd used hybridize to the 5' end of the message, but it hybridized at 2 different places. And so there were 3 pieces of RNA all being made by adenovirus that were being joined together to make the mature message. And this was the discovery of split genes. Now, this was done on a Tuesday morning. On Tuesday afternoon, people were already talking about Nobel Prizes. Everybody was very excited about this. We went out, talked to all our friends about it, and then went on and did the necessary work in order to write it up as a paper. This was the paper that came out in 1977, and the title was An Amazing Sequence Arrangement at the 5' ends of adenovirus 2 messenger RNA. We already knew at that time that there were many eukaryotic RNAs that showed exactly this same thing, because as soon as we started talking about this, people would say, oh, I can now interpret my experiments. We had one situation at a Cold Spring Harbor symposium where I talked about the discovery at the start of the meeting, and Mike Bishop, who later got the Nobel Prize for the discovery of oncogenes, said, oh, now I can explain this slide that I brought along with me that I couldn't explain before. And so it was immediately accepted. Everybody had all the results, and our results were very clear. So anyway, we sent this paper in, and there were 3 reviewers, and all 3 reviewers wrote back and said, well, you know, this is an excellent paper and so on, and they liked everything, a few minor points. But every one of them said they didn't think it was appropriate to use the word amazing in the title of a paper. Well, I had spent a lot of time figuring out what was the best word for this. And so I called up the editor of Cell, Ben Lewin, and started talking to him about these results that we got, and particularly the criticisms we got from the reviewers. And during the course of the conversation, I casually asked him, you know, when you first heard about this, what did you think? And he said, oh, it was amazing. I said, I rest my case. And so that's how the word amazing got into the title of this paper, which, as far as I know, is the first time that it was ever used in the title of a scientific paper. So that was good. And everything went very well. In 1993, I got the Nobel Prize along with Phil Sharp for the discovery of split genes. Phil had done similar experiments at MIT, but completely independently. He didn't know so much about this. And in fact, we got the results slightly before he did, although he rushed to get his paper out to try to get precedence. There were a lot of other people who also published in 1977 trying to claim that what was going on in viruses was different, and that what was going on in eukaryotes, in higher organisms where splicing was everywhere, that this was kind of different. But fortunately, the Nobel Committee decided that it was Phil and I who should share the prize. Now, one of the things you have to do when you go to Stockholm for the Nobel Prizes is to give a lecture. And almost everybody talks about all of the clever experiments they've done since they won the Nobel Prize, and talks about the experiments that followed up on the discovery. Now, we had started back in 1977, 78 time, trying to set up an in vitro system where we could study splicing, but we had not been very successful. And it turned out I just picked the wrong promoter and the wrong RNA polymerase to try to make the RNA in vitro. And Tom Maniatis and Adrian Craner had done a much better job, and they in fact got way ahead of us. And so after a little time, we stopped working in this area and started working some more on restriction enzymes and on the DNA methyltransferases that covered them. And in 1993, about 2 months before I had to go to Stockholm, with Xiaodong Cheng, a crystallographer at Cold Spring Harbor, we discovered that if you take the DNA methylase, HHA1, which recognizes GCGC, and you bind it to DNA, the base that is going to be methylated is flipped right out of the helix and into a pocket in the enzyme. And that was a very exciting discovery. We now know that this is very common, that many, many enzymes that interact with bases in DNA do exactly the same thing. And so this was quite a big discovery. And this was what I talked about for my Nobel talk. And of course, you know, if you make a nice discovery, then it's always good to make sure the Swedes know about it. So, so far, nothing more has happened from that. Now, as I told you, back in the 1970s, 74, 75, we had been discovering a lot of restriction enzymes in my lab. And so one of the things that I wanted to do, we'd been giving these away. People would come to meetings at Cold Spring Harbor and they'd have DNA in their pocket, and they'd come down during a break and say, do you have a restriction enzyme that will cut my DNA? And it was obvious there was a business here, that one could make these enzymes and sell them. And so that's what I tried to do. I tried to persuade Jim Watson that we should set up a company at Cold Spring Harbor to sell the restriction enzymes and use the profits to support the research at Cold Spring Harbor. Jim didn't like that. As I say, we fought a lot, and that may have had something to do with it. But he said, oh, you can't make money doing this, and it's kind of boring. And he didn't think it was appropriate for an academic lab to get involved in any sort of company. But I knew that there was a business here, and I really was fed up of just making these enzymes and giving them all away. And so I heard through a friend at NIH that there was a company called Miles Laboratory, who at the time were kind of the Sigma of the world, and they were selling reagents that they purchased from other people. And they said, my friend said, call Miles and see if they would be interested. So I called them, and they said, well, it's not us. We've been thinking of selling one, but this man, Don Combe, shown here in the picture on the left, is in Boston, and he's been making one enzyme for us that we're going to sell. Why don't you talk to him? So I called him up, and we went on. He said, oh, I told him I had 30 enzymes that we discovered at that time. And then he said, come up, let's talk. I get up to Boston, to Beverly, actually, just north of Boston, and discover he's got a little lab going. He, his wife, and one technician, they have a makeshift lab in the basement of a hairdressing salon in Beverly. And he'd been making some reagents, tRNAs, iso-accept tRNA synthetase and so on, and had come across this idea of making what endonuclease are that I'd heard about from Dan Nathans. And so I said, well, I've got 30 more. And he said, oh, that's good. And he was very keen to do it, but he was thinking about selling them via Miles and not selling them directly. And so I convinced him that we should try selling them directly. And he offered me a partnership in the business, which I didn't want. And so I said, well, I'll just be your chief scientific advisor. And so, and then I can continue to do my work at Cold Spring Harbor because we hadn't yet discovered RNA splicing. So he said, fine, that's fine. And I gave him a list of all the people who kept coming to my lab wanting free restriction enzymes. And we developed a mailing list. I put together a sort of primitive catalog of what was possible, sent him the strains, told him how to make them. But I made it clear that I wasn't going to enter into an exclusive arrangement with him. If other people wanted the strains, I would pass them out. And we had no patents or any. This was before it became really important to get patents. Anyway, the bottom line was that in the first year, starting in 1975, Don Combe and New England Biolabs made $200,000 selling restriction enzymes directly to customers. And we have continued that tradition today. We are now a company of about 650 people. We moved in 2005 to company headquarters in Ipswich, Mass. It's about 30 miles north of Boston. We have distributors around the world, France, Japan, Singapore, UK, and so on. And we do a rather large amount of business. We sell a lot more than restriction enzymes, including enzymes that are necessary to modify messenger RNA. So the mRNA, the methylases that are necessary, the RNA methylases that are necessary to put the cap structure and modify it on messenger RNA, we made. And so Moderna and their vaccine all came using reagents that we'd made here at New England Biolabs. So that's been really a very successful story. And I think it illustrates well how it can be very useful to get involved with industry when you realize that you have reagents that can be useful. Let me tell you a little bit about things that have happened since I won the Nobel Prize. I've always been fascinated with puzzles and games, and I spent my Nobel Prize money putting a professional croquet lawn in front of my house. That's me at the bottom left, sitting on the lawn. I also like fun. I like jokes and so on. And if any of you have heard of the Ig Nobel Prizes, these are something that are sort of the opposite of the Nobel Prizes. They're intended to be experiments or work that's been done that sounds funny, but then when you start to think about it, you realize that it's very serious work. I'd learned to play croquet when I was at the MRC lab in Cambridge. And it's nice because it combines the skill of snooker, in which you're trying to pop balls, which is also something I loved, and with a strategy of chess. And so it's a very intellectual game, much more so than many of the sports that one gets involved with. I also got a number of honors. I've been awarded a number of honorary degrees of one sort and another. The science department at my old grammar school was named after me. The expansion of the chemistry department, the University of Sheffield, was named after me. I was knighted actually by Prince Charles, now King Charles, in 2008. And I've also been very active in organizing the Nobel laureates to try to do good things, either to correct science that is being misused or for humanitarian purposes. And one thing, I helped organize the Nobel laureates, getting Bulgarian nurses released from jail in Libya, where they'd been charged with deliberately spreading HIV through the children's ward in Benghazi. Now, I want to finish by talking about several things. One, I think everybody needs to look back from time to time and think how important luck is. And I learned this when I was about 18. I thought I was going to get thrown out of school because I stopped going, and I used to spend my time playing billiards and snooker. And at one point, even thought about being a professional snooker player. But I was in a tournament one day, and an old gentleman came up to me afterwards, and I had made a lucky shot during the course of my play. But then after having made the shot and being able to continue my break, I messed up the next shot. This old gentleman came up to me and he said, you know, when you have luck, the most important thing is that you take advantage of it. Make sure that if you've got some lucky break, you do everything you can to capitalize on it. And I found this very important because an awful lot of people are lucky. Everybody has luck, but a lot of them feel guilty about it. They really don't, you know, they don't take the necessary steps to take advantage. And you should take advantage. It's very important. So that was a lesson I learned very early on. Kazu Kurosawa, it was lucky, quite by chance, I was put in a lab in which he was a postdoc and became my best mentor ever. I ended up at Harvard, rather Wisconsin, as a postdoc, just because Jack Strominger had recently been appointed a professor at Harvard. Looking for eukaryotic promoters in adenovirus, we didn't know what we were going to discover. We thought that it was a relatively simple experiment that we could compare promoters in eukaryotes and in bacteria. But perhaps the luckiest thing that ever came is taking an early plane. Many of you will have heard about the crash that took place when some of Osama bin Laden's people crashed a plane into the first World Trade Tower in New York. I was booked on that plane, the one that went into the World Trade Tower, until 2 weeks before it took off. And I'd been going to a meeting over in California, and the meeting was moved forward by a day. And so I ended up taking that plane, not on September 11th, but rather on September 10th. And that is luck. Had I been not so lucky, I would not be here to talk to you today. And the last thing I want to mention is studying bacterial RM systems. This was, again, luck. I started doing it because I heard a lecture from Dan Nathans. Jim Watson wanted me to do something else, and I didn't think that was a good idea, and I took advantage of the restriction modification systems, led to many, many papers, led to the formation of New England Biolabs, and really has had a huge impact on my life. I now work at New England Biolabs. I've worked there since 1992. I study DNA methylation, and it turns out that DNA methylation is very important. It's not just about restriction modification. It actually, the methylases control a lot of biological activity, and we often don't know what that is. And there are still many discoveries to be made in that field. I'll stop at that point. I'll thank you all very much for listening, and I will be extremely happy to answer any questions that you might have.
Dear Professor Roberts, thank you very much for this fascinating lecture. Actually, it's really very, very useful for all researchers, but for young researchers especially. I would like to note that it's really amazing that you succeeded in convincing the editor of the journal to keep the word amazing in the title of the paper. Well, now I ask students who would like to give a question. In the questions, please. Well, they think about. So in the virtual space. I just look, I do not see in the chat. Well, you told the story of your career, and it's really fascinating. And I think that luck comes to people that deserves it. That's one issue. And the second, of course, passion and curiosity, drive, and activity led to winning the Nobel Prize. It's of course impressive, and as you told, this curiosity appeared from the times when you were pupil, like we have now here in the blue hall of the academy. So I again ask students and pupils, in case there is a question, please formulate your question.
Honorable Professor, my name is Eugene Valik. I'm a fifth-year student of University of Medicine and Pharmacy Nucleitis Timișoara, and I have a question to you. Which books can you recommend to us in order to achieve such great and big success in research and eventually a Nobel Prize? Thank you.
Well, you know, part of the problem there is that there are a very large number of books that are worth reading. There's a very good book called The Molecular Biology of the Gene, which was originally put out by Jim Watson and several other authors, and it has been updated a number of times since then. That gives a very good overview of where we are with molecular biology at the moment. But you also have to realize that it is moving very quickly. And I always recommend to students, rather than focusing on a book, because they're almost always out of date as soon as they're written, is to read, assiduously read, really go through journals like Science and Nature, where you will learn about the very latest developments in a number of fields, not just the one you're working in. And I also recommend reading books that are outside of your field. I've just read a very, very good book, I've not quite finished it yet, about women in science. It's called The Exceptions, and it talks about the difficulties that women faced when they were trying to become scientists back in the 1960s and 1970s, but which they also face today. It really is about a lady, mostly, who is a professor at MIT. Her name is Nancy Hopkins, Hopkins, and it really is just wonderful in explaining the difficulties and often how to overcome them that women have faced. And one of the things that comes through is it is very important to be activist. Don't sit back and let people push you around. Be activist. If you see that something's going on that is wrong, bring attention to it. Try to do something about it. And I've tried to do that with these campaigns that I run with Nobel laureates. We've run now about 40 campaigns and have been quite successful on some of them. But it's because we're not satisfied with what is going on.
Okay, thank you so much. We have a question in the chat. Slav Tatiana, so have you ever regretted that you changed chemistry to molecular biology?
Well, you know, to ask a Nobel Prize winner in molecular biology that question is very difficult. Do you think I might have won 2 prizes if I'd stayed in chemistry? I don't think so. No, I never regretted it at all. And in fact, the basics of life are all about chemistry. You know, when enzymes work, when DNA gets replicated, when RNA gets made, this is all chemistry. It's just the underlying chemistry that is so important. And so in one sense, I don't think I left chemistry. I think I've just embraced a better type of chemistry, if you like. And I would also note that because all of the work, almost all the work I do is bioinformatics, I also used to program for a while. And so my love of mathematics has been very important here. And of course, I'm also a detective. So everything I planned on becoming when I was a kid, I feel I've really ended up becoming that. And that is a good feeling, a very nice feeling. So no, I wouldn't change a thing.
Yeah, dear Professor, your father created some possibilities to develop your curiosity, let's say, or to satisfy your curiosity. Now I would like to ask you concerning your children or your students, is the same approach from your side?
Well, you know, my children, 3 of them avoided science. 1 of them is interested in computer programming. He's a professional programmer, and I view that as science. But fortunately, I have 3 grandchildren, 1 of whom is actually working as an intern at my company this year at New England Biolabs. And he is doing very well. I think there's every chance he will become a scientist. And I also have a granddaughter who's about to go to college, and I'm hoping she too will become a scientist. So the first generation didn't do so well, but perhaps the second generation will do better. But I think it's really important, you know, for all kids, they're all curious, they're all natural scientists. They have questions, they want answers. And I think we have to be very careful when we educate them in schools that we don't knock that curiosity out of them. We must make sure they have the opportunity to find out what is going on in this world.
Yeah, dear Professor Richard Roberts, your lecture was very interesting and very useful. We have pupils from lyceums, students, talented young people, and I am sure that they enjoyed your presentation. Thank you so much. Now we will pass to